and welcome back to the Social Neuroscience Network. In today's video, we're going back to the basics. We will review some of the key things that I think you need to know about neuroscience before we dive more into the social brain in adolescence. We will review what neuroscience is, divisions of the brain, neurons and what you need to know about them, and action potentials. Ready? Let's go! So first, let's start with the most basic of basics. What is neuroscience? Neuroscience is the study of the nervous system. The nervous system can be separated into two parts, the central nervous system, referred to as the CNS, and the peripheral nervous system, referred to as the PNS. The CNS consists of the brain and the spinal cord, and the peripheral nervous system consists of the network of neurons. Neuroscientists study the nervous system to answer questions varying about its functioning and how it contributes to the manifestation of various behaviors and neurological disorders. It is a broad field with many concentrations such as cellular and molecular neuroscience, behavioral neuroscience, computational neuroscience, translational and clinical neuroscience, and sensory system neuroscience, to name a few. Let's turn to the main character of our story, the brain. The brain is one of the largest organs in our body and is also the most complex. The brain can be divided into three main divisions, the cerebrum, the cerebellum, and the brainstem. The cerebrum consists of an outer layer of gray matter called the cortex and an inner layer of white matter. The cerebral cortex can further be divided into 5 lobes: the frontal lobe, the parietal lobe, the occipital lobe, the temporal lobe, and the insular lobe. The frontal lobe is the largest lobe in the cortex and is located in, you guessed it, the frontal part of the brain. The frontal lobe has many roles. It's involved in movement, cognitive control, self-referential thought, and executive functions such as planning, organization, and goal-oriented behavior. Next in the cerebral cortex is the parietal lobe. The lobe is located sort of in the middle of your brain, posterior to the frontal lobe and anterior to the occipital lobe. The parietal lobe is also involved in multiple functions. This lobe has a role in sensory motor planning, somatosensory functions like touch, pain, temperature, and proprioception, learning, language, stereognosis, which is the ability to tell the difference between objects based on size, shape, and weight, and it's also involved in multisensory functions like spatial processing. Anterior to the parietal lobe is the occipital lobe, the smallest lobe in the cortex. When you think occipital lobe, think vision. The primary role of the occipital lobe is visual processing, containing both the primary and association visual cortex. Speaking of sensory systems, that leads us to the next division of the cortex, the temporal lobe. The temporal lobe is located on the left and right sides of the brain. The most prominent role of this area has to do with the auditory system. That being said, the temporal lobe is also involved in other essential functions, such as object and face recognition, processing emotions, memory, and language. Last but not least, the insular lobe. There's a scarcity of functional data on the insular lobe in humans, and this all comes down to its location in the brain. The insular lobe is hidden under the temporal, frontal, and parietal lobes, forming the floor of the lateral sulcus deep in the brain. The lack of data results from the lack of accessibility of this region. That being said, we still do understand some of the functions of the region. The insular lobe is believed to be important for taste and sensory motor processing, auditory and vestibular functioning, risk-reward behavior, pain pathways, and autonomics, functions that are involuntary and related to the autonomic nervous system. Okay, so that's the overview on the cerebral cortex. Next division of the brain on the list is the cerebellum. The cerebellum is located between the cerebrum and brainstem and is one of the cutest regions in my opinion. The white matter in the cerebellum, referred to as the arbor vitae, makes it look like a little tree. In fact, arbor vitae in Latin means tree of life. How fitting is that? The cerebellum's main responsibility is movement. The cerebellum is involved in movement regulation through the coordination of gait, posture, muscle tone, and voluntary muscle activity. However, it cannot initiate muscle contraction. That's reserved for the motor cortex. The cerebellum also plays a role in balance control. Damage to the cerebellum in humans results in the loss of ability to control fine motor movements, maintain posture, and motor learning. Last, this brings us to the brainstem. The brainstem stems from the bottom of the brain and connects the cerebrum to the spinal cord and cerebellum. The brainstem is responsible for maintaining the vital functions of life, such as breathing, heart rate, blood pressure, and consciousness, to name a few. The brainstem is essential. Without it, the brain can't communicate with the rest of the body and initiate behavior and action. Okay, so that's the big picture of the brain. Let's take out our microscopes and zoom in to a more molecular level. What do we see? Neurons. Neurons are the building blocks of the brain. If the brain is an oddly realistic cake, nerve cells are the ingredients that make it. Neurons are the nerve cells in the brain that are responsible for receiving and sending messages between the body and the brain. These signals are called action potentials, something we'll discuss in a moment or two. The neuron is comprised of three main things: the cell body or the soma, dendrites, and axons. The cell body or the soma contains the nucleus of the neuron. They also carry the required proteins for the synthesis of enzymes or other molecules essential for cell life and functioning. Emerging from the cell body like bushy branches are the dendrites. Dendrites provide the main structure where the neurons receive incoming signals from another neuron. Extending from the cell body and forming a pathway is the axon. Axons are typically longer and thinner than dendrites and provide the pathway with signals that travel from the cell body to other neurons. The membrane along the axon is designed to propagate action potentials. Emerging from the end of axons are axon terminals. Forming at the end of axon terminals are something called terminal buttons. Terminal buttons are information delivery portion responsible for sending the signal off to other neurons. How? Great question. Terminal buttons contain something called synaptic vesicles. Synaptic vesicles are small organelles that store neurotransmitters. Neurotransmitters are endogenous chemicals that are what allow neurons to communicate. At the arrival of an action potential to the terminal button, calcium-evoked exocytosis causes these synaptic vesicles on the presynaptic nerve terminal to discharge their neurotransmitters into the cleft between this terminal button from the dendrite of another neuron. This cleft is called a synapse. Here, the neurotransmitters bind to the postsynaptic receptors, which begin the receiving of signals on another neuron. Let's dive in a little bit more on synapses. Synapses are the point at which information from one neuron can be transferred to another neuron. Synapses are often made between the axon of one cell and the dendrite of another, forming a sort of communication zone where information from one neuron can relay to another. However, this does not always have to be the case. Sometimes synapses can be formed between axons, between dendrites, and between the axon and a cell body. However, the most common synapse formation occurs between the axon of one neuron and the dendrite of another. Synapses can be excitatory or inhibitory. Excitatory synapses tend to promote the firing of another neuron. Inhibitory neurons can cancel the signal that would otherwise excite the firing of other neurons. Who regulates whether synaptic transmission is excitatory or inhibitory? The type of neurotransmitter and the postsynaptic receptor they bind to. There are 8 main neurotransmitters. These are acetylcholine, glutamate, GABA, histamine, epinephrine, norepinephrine, serotonin, and dopamine. The main excitatory neurotransmitter is glutamate. Epinephrine and norepinephrine, often referred to as adrenaline and noradrenaline, also have excitatory effects. The main inhibitory neurotransmitter is gamma-aminobutyric acid, or, more simply put, GABA. Dopamine and acetylcholine can have either excitatory or inhibitory effects, depending on the receptor they bind to. The third category of neurotransmitters is the modulatory neurotransmitters. This includes neurotransmitters such as histamine, dopamine, acetylcholine, and serotonin. To recap, the brain is made up of nerve cells called neurons that are responsible for sending and receiving signals, also known as action potentials, throughout the brain. To make it simple, I like to think of them like little gossipers. The dendrites are the eavesdroppers and pick up on a conversation being had by a neighboring neuron. The dendrite turns and whispers a secret to the axon, who tells the terminal button, who cannot keep her synaptic vesicles shut, and blurts it into the synaptic cleft, where the rumors and secrets either spread to the next gossiping neuron or are shut down / inhibited, terminating the signal. However, unlike the rumors spread in your high school homeroom, these messages are a bit more reliable. Oh, also, just an FYI, there's another type of neurocell in the brain called glia. We don't really have enough time to go into too much detail on these, but know that glia essentially occupy the space in the nervous system not taken up by actual neurons. Glia provide structural and metabolic support for neurons. For example, transporting nutrients to the neuron or clearing up excess neurotransmitters. The neurons are the UFC cage fighter, and the glia are giving it water and wiping up sweat and blood on the cell lines. All right, back to the neuron. I've thrown around the term action potential quite a bit in this video. Right now, we know it's the signal communicated between these neurons, but I haven't gone into just how that happens. First, we need to set the stage. The membrane of the neuron, about 5 nanometers thick, tiny, I know, has 2 layers of lipid molecules that are arranged so that the hydrophilic ends, which are attracted to the water, are pointed towards the water, inside and outside the cell, to form the exterior neuronal membrane, and the hydrophobic ends, which are resistant of water, are facing away from the water, forming the interior neuronal membrane. The membrane establishes both an intracellular and an extracellular matrix for the neuron, which is incredibly important for action potentials. The intracellular and extracellular matrices have different distributions of ions. Sodium, calcium, and chloride ions concentrations are higher in the extracellular matrix than the intracellular matrix, whereas potassium concentrations are higher in the intracellular matrix than the extracellular matrix. As you may recall from your chemistry class, ions have a net electric charge resulting from the loss or gain of electrons. The charge is indicated in the superscript above the element. For example, sodium, written Na+, loses an electron when becoming an ion, leaving it with 10 electrons and 11 protons, so the ion carries a positive 1 charge. Chlorine, on the other hand, gains an electron, making the distribution 18 electrons and 17 protons, so we walk away with a negative 1 charge. The charges and distributions of ions become important when we talk about the membrane potential. The membrane potential is the difference in electrical potential inside the intracellular matrix relative to the extracellular matrix. When a neuron is not excited or resting, we have something called a resting membrane potential. The resting membrane potential in a neuron is about negative 70 millivolts. Embedded in the membrane is a sodium-potassium pump, which also aids in maintaining the membrane potential in these neural cells. Ions tend to move spontaneously across from a region of high concentration of the specific ion to a region that has a lower concentration, also known as moving down a concentration gradient. As we mentioned, the extracellular matrix has a higher concentration of sodium than the intracellular, whereas potassium concentration is higher in the intracellular matrix than the extracellular. So, according to the concentration gradient, sodium is going to want to naturally flow into the cell, and potassium is going to want to flow out of the cell, moving from an area of high concentration to low concentration. But wait, that's going to mess with the membrane potential. We can't have that. Well, that's where the pump comes in. The sodium-potassium pump moves these sodium and potassium ions against the large concentration gradients in order to maintain the resting membrane potential. This is crucial for the regulation of cell signal transduction, which we will go into momentarily. The pump uses ATP for energy. For every ATP the pump consumes for energy, the pump will pump 3 sodium ions out of the cell and 2 potassium ions into the cell. This process ensures our membrane potential stays around negative 70 millivolts at rest. So why is this important? I'm so glad you asked. Changes in the membrane potential is actually how neurons communicate. Action potentials, the signal arising from one neuron, sending information down the axon to communicate with another neuron, is a rapid change in the voltage across the membrane. Action potentials look like the chart shown on the screen. Action potentials occur in 3 stages: depolarization, repolarization, and hyperpolarization. Let's break this down. When a stimulus occurs, voltage-gated sodium channels open, and sodium begins to influx into the cell. Sodium ions carry a positive charge, so when they enter the cell, the negative membrane potential begins to move towards 0 millivolts. The membrane voltage is becoming less negative, and this is what we refer to as depolarization of the cell. Once the potential hits negative 55 millivolts, the threshold of the cell, the neuron fires an action potential. If this threshold is not reached, no action potential is fired. This is what we mean when we say action potentials are, quote, all or nothing. The threshold is either met and an action potential fires, or it isn't and nothing happens. At the firing of the action potential, the voltage-gated sodium channels close and voltage-gated potassium channels open. However, voltage-gated potassium channels are kinetically slower than sodium channels, so this opening occurs about 1 millisecond after the closing of the sodium channel. Once the potassium channels open, potassium ions rush out of the cell, or efflux. These positive ions rushing out of the intracellular matrix begin to reverse the effects of depolarization as the voltage begins to become negative again. The flow of potassium ions out of the cell and decreasing the membrane voltage back towards the negative 70 millivolt resting potential is referred to as repolarization. Once the membrane potential falls back below the negative 55 millivolt threshold, the potassium channels will begin to close. However, the slow kinetics of this channel results in the voltage-gated potassium channels not closing immediately after threshold is hit. This delay in the closing of the potassium channels results in more ions effluxing than necessary, and the voltage will dip below the negative 70 millivolt resting potential, becoming more negative. This period of dipping below the resting potential is referred to as hyperpolarization. Eventually, the natural permeability of the neuron to sodium and potassium ions will restore the ion concentrations back to resting levels, and the resting potential is reestablished. All right, that about does it. In today's video, we got introduced to the field of neuroscience, got a brief overview on the divisions of the brain, covered the structure of neurons and how they communicate. Now, this is in no way an all-encompassing review. Many spend months going into much deeper detail on the topics discussed today. That being said, I hoped this provide a good foundation for you as we begin to dive deeper into the special interest topics. Thanks for tuning in, and we'll catch you on the next one. Bye!